Hello. Today I'll be discussing immunohistochemistry, which is a molecular process and technique with many beneficial uses that will be outlined in this presentation. Before beginning, it is important to understand the word immunohistochemistry. This understanding comes from the stems within the word. As shown on the slide, immuno means that it is in relation to the immune system, and therefore with immune response. If you think back, immune responses involve the production and binding of antigens and antibodies. Both of these are proteins and will be highlighted further along in this presentation. Histo is dealing with tissues, therefore the material being analyzed with immunohistochemistry is the isolated tissues found within a body or organism. The overarching goal of immunohistochemistry is to tag a tissue for the location of where a specific protein may be within a sample. Before continuing to outline the process that occurs when doing an immunohistochemistry assay, there are multiple different uses for the process that are important to outline. First, it can be used for the diagnosis of a condition, such as Alzheimer's disease, Parkinson's disease, or muscular dystrophy. Immunohistochemistry can also be used to determine a prognosis for a specific disease and to measure and predict how the individual is responding to a treatment for a disease. Now, the process of immunohistochemistry will be described. First, a tissue of interest is extracted from the organism and prepared to be in an isolated form with no contaminants. At the same time, molecular processes occur to prepare antigen binding sites on the surface of the tissue. Next, a primary antibody is added to the tissue that can bind to the antigen binding sites. Both antibodies and antigens are proteins, therefore showing the prevalence of such proteins in a tissue. Antigen binding sites are specific for an epitope or shape of the antibody. Only the specific antibody can bind, resulting in the target protein being revealed. In order to visualize the protein, a secondary antibody is added to the mixture that carries a fluorescent tag. If the correct binding occurs for the target protein, the fluorescent tag will show exactly where the protein is expressed in the tissue. If the target protein is not present in the tissue, the antibodies will be washed away during the washing stage that occurs after each incubation, which is directly after each antibody is added to the solution. Immunohistochemistry is a molecular process that was described in a recent publication by Victoria Mott, PhD with the University of North Carolina at Chapel Hill. In this publication, the effects of adolescent intermittent alcohol exposure, shown as AIE on the slides, has on the brain of rats. In this study, the different proteins within the brain that are responsible for spatial learning as well as reversal learning was examined. Immunohistochemistry was completed on the brain tissue of rats in the study, showing the expression of a protein present in developing neurons. Therefore, the alcohol, or AIE exposure, can be drawn back to immature neurons, when normally these neurons would not still be in development in an immature stage. Lastly, the immunohistochemistry was used to show the increases or decreases in certain learning proteins within the brain after the AIE exposure. Thus, it is shown that immunohistochemistry is a valuable assay in the scientific world. Shown on the slide are the resources used to gather information shared in this presentation.